Hi, I'm Linda van Aelst, a professor at Cold Spring Harbor Laboratories, and research in my lab has been focusing on the role of role regulators in different aspects of neuronal development and function and its implication in disease, as mutation in genes encoding such regulators have been implicated in human neurodevelopment disorders, including mental retardation, autism, and schizophrenia. We have been focusing particularly on one such regulator, Oligophrenin. Mutations in the gene have been reported in families with mental retardation. In a previous study, we demonstrated a role for Oligophrenin at the hippocampal CA3-CA1 synapse during early development in glutamatergic synapse development. Excitingly, in a more recent study, we, Neil Nadev Kasri and Akika Kobayashi, also found an important role for Oligophrenin in a form of plasticity that requires the activation of metabotropic glutamate receptors, in short, mGluR-LTD. A key feature of mGluR-LTD, which in the hippocampus can be induced by either mGluR1 and mGluR5, is the reliance on local de novo protein synthesis. The prevailing model, in fact, is that mGluRs trigger the local translation of proteins, referred to as LTD proteins, to cause LTD by enhancing the rate of AMPA receptor endocytosis locally at active synapses. Its importance in cognitive impairment has been demonstrated by the findings that this form of plasticity is altered in a mouse model of Fragile X, which has led to new therapeutics for Fragile X syndrome acting on mGluR5. However, key remaining questions are: what is the nature of these locally synthesized proteins, and are there other cognitive disorders that are associated with alterations in mGluR-LTD? In biochemical studies, we had found an interaction between Oligophrenin and proteins that had been implicated in mGluR-LTD. This led us thus to explore as to whether Oligophrenin could play a key role in mGluR-LTD. We first tested whether OPN protein levels are rapidly induced upon mGluR activation, and we found that this is indeed the case, and interestingly that the rapid upregulation of Oligophrenin depends on mGluR1 rather than mGluR5 and is not dependent on FMRP regulation. To demonstrate that Oligophrenin is required for mGluR-LTD, we relied on RNAi interference and demonstrated that acute blockage of mGluR-induced Oligophrenin synthesis impairs mGluR-LTD. Furthermore, we demonstrated that Oligophrenin mediates mGluR-LTD by enhancing AMPA receptor endocytosis via interaction with core components of the clathrin-dependent endocytosis machinery, endophyllin 2 and 3. Thus our findings identify Oligophrenin as a key molecule playing a role in mGluR-LTD. Importantly, its role in mGluR-LTD can be molecularly dissociated from its role in glutamatergic synapse development. Whereas its role in mGluR-LTD requires interaction with endophyllin 2-3, its role in glutamatergic synapse development requires its role gap activity and its interaction with the scaffolding Homer1 BC proteins. So we are excited about these findings, as such Oligophrenin on the one hand can play an important role in synapse maturation and circuit wiring during early development, and on the other hand, regulated Oligophrenin synthesis can operate during adulthood to weaken the synapses in response to behavioral relevant stimuli.